Rightio, let's make a start. Good morning, everybody. For us in the southern region, they're forecasting rain this weekend, which is fantastic. So I hope everybody gets rain that needs rain. But going and talking about the crop, today we've got a presentation on nematodes and sugarcane. Question is, are they friends or foe? And how much work has been done, or how much do we know? So I'm James Ogden-Brown. For those that don't know, I'm the regional coordinator based at SRA in Bundaberg, and will be your co-host for this morning's webinar. The webinar presentation will be presented by Dr. Shamsul Bhuiyan, Principal Research Scientist in Pathology, to showcase SRA's research on management of plant parasitic nematodes for over the past 10 years. So a little bit of housekeeping. Please note that today's webinar is being recorded. We encourage you to keep your microphones on mute to keep the background noise to a minimum. If you're having any technology issues, please message myself or raise your hand and we can assist where possible. We do encourage everybody to take an active part in today's webinar, but please can I ask you to refrain from asking questions during the presentation. So we'll ask for questions at the end. You can either put it in the chat box or raise the hand on the screen here and ask your question verbally. So thanks everybody, and thanks to Shamsul, who I'm now going to hand over to you. So over to you, Shamsul.
Good morning, everyone. Hope you have a good, nice sleep last night. So today's presentation, as James mentioned that we're going to showcase SRA's research on sugarcane nematodes. So we have started this work in 2011, and then since then we have done a fair bit of work. This is my attempt to actually show you what we have done last 11 years. So let's start. So this is the outline of my work. So what we're going to talk, because of it's a sugarcane nematode, but we have to give you a bit of an overview actually what are nematodes. I think most of you know. And then I'll be talking about the plant parasitic nematode of sugarcane. And then I will be talking about the nematode survey we have done done a few years ago. And then
Shamsul, could I just interrupt? If you'd like to, could you put it on presentation mode? We're just seeing all of your slides. So yeah, if you hit slideshow. Yep, brilliant.
Is all right now?
Yep, great.
Sorry about that. Yeah. So, and then I'll be talking about the management options. So I'll talk about actually different management options for nematode. But in particular, the work we have been done for last 10 years. And at last, we'll be talking about the nematode community, how we can use the nematode community analysis as an indicator of soil health. So let's move on. So what are nematodes? So they are mostly microscopic animals. They are long, worm-like animals, multicellular. They live almost everywhere in the world. You can go into the Gobi Desert, you will find them. You can go to the Himalaya, you will find them. And then it is in the tropical islands, so almost everywhere, even in the bottom of the sea. So in soil, we usually find two types of nematode. One we call is free living. The other types of nematode we call parasitic. So what is parasitic? Sorry, what is free living nematode? So the free livings, actually, they're not actually living free. They have to actually feed on something. So they don't actually feed on plants. So they feed on bacteria, fungi, algae, and other nematode and the organic matter. But in doing so, they are actually the important part of the soil ecosystem. And they play vital roles in nutrient transformation and decomposition of the organic matter. So as I mentioned before, that the nematode, this community of the nematode can be used a vital information about the health of the soil, which I will be talking at the last of my presentation. So what are the plant parasitic nematodes? The plant parasitic nematodes are small worm-like organisms which mainly lives in soil and then attack roots. But some of the nematode, they could actually attack the upper part of the nematodes as well, say for flower and fruits. This is I think the plant parasitic nematodes is a huge problem for food security for the world, because if you get all the horticulture crop and agriculture crop, nematode actually cost around $150 billion loss of productivity. So now we come to the sugarcane nematodes. So as we know that sugarcane is a perennial crop, so that means that it lives for more than 1 year. And then for this reason, it has actually most diverse community of nematode in sugarcane soil. About more than 300 species lives in sugarcane soil. So, but the most important parasitic nematodes in sugarcanes are root knot and root lesion nematode, which I'll be talking today. So there are a few other nematodes are as well. So they are typically minor, but they could create a major problem if they actually present in large number. So, but in general, they are not important nematodes for sugarcane. So, as I say, that root lesion nematode is one of the most important nematode of sugarcane. The reason is, I think it has the wide adaptability. It can occur in all types of soil and all types of environment. So if you look at the sugarcane soil, you can find in every region. And the other thing is, it is an endoparasite. What does it mean? That it can actually dig a hole into the plant roots, and it's actually destroy the cortical roots, cortical tissue, and then it lays eggs, and the eggs actually hatch, and then the juvenile coming out of the roots, and then actually they find the new root and then attacked it. So as a whole, at the end, you don't find any actually fine roots in sugarcane root system. So the other one is root knot, which is mainly restricted to the sandy soil, but because it's actually happening in the sandy soil, but it is actually the most damaging parasite of sugarcane. What it does actually, it attacks the root tips of sugarcane root. And what happened so it produces gall, and it's actually stopped the elongation of the roots. If you can look at this picture, you'll see the left side, actually the attacked root with gall, they are shorter and stunted. If you look at the right side, you see the roots are much healthier root. And if you look in the same plant with different result, just for the root attack of nematode. So altogether, these two causes Australian sugar industry more than $80 million loss of productivity every year. So you can see that, as I said, that it's actually causing 80 million year loss of productivity. And if you look at the percentage of crop loss in Australia, about 10 to more than 20%. So it's not only in Australia, it's actually in other countries as well. Whoever actually dig into the soil to look for nematode, they find it, and then if you actually look for it, you'll find actually it's actually causing damage. It's actually, nematode is like kind of RSD or some other diseases. It is subtle. You can actually, there is no symptom. That's the problem. But if you look at it, and if you actually do the measurement, you can see really it actually do lots of damage to sugarcane crop. So now I'll be talking about the survey work we have done last few years. So the first nematode survey work been done in 1980s, 1990s under the sugarcane yield decline joint venture project. And they actually set a foundation of nematode research in Australia. So they first time they found that the nematode is a problem for the Australian sugar industry, and it's present almost everywhere across the sugar industry. And also they estimated a yield loss that it can actually cause substantial damage or impact the productivity. So but at just a sort of like we are thinking, what can we do actually? Because after this work been done 20 years ago, and then after that, nothing been done. So we wanted to do some actually test run survey. So we, in collaboration with Sunshine Sugar in New South Wales, and in collaboration with HCPSL in Harvard, we conducted a survey from 2018 to 2020. So what we did actually in the survey, instead of putting the nematode number, we wanted to see actually the nematode present in this area pose any risks to the productivity. So what we did, we calculated a index we call is nematode hazard index. What does it mean actually? So we know that the nematodes have, even the mild nematode, pathogenic nematode can actually do significant damage if they actually present in high number. So we arrange this nematode in a range of to their relative pathogenicity. So we know that root knot nematodes is a highly pathogenic, so we put them at the hazard index of 1, and then relatively we put the other nematode as. So for example, if you have a root lesion nematode, we can say that it actually does the 80% damage, then it would cause damage by the root knot, so on. So what we did after collecting the nematode, and then we made the hazard index. So the good thing about the hazard index, we can actually relate to the risk, and then can be actually related to the yield loss. So for example, if you have a risk category, it is actually false. Risk category low, we can see the yield loss could be insignificant, but if it's actually in the risk category high, we can say that the potential risk of yield loss is very significant, about more than 20%. So this is the result of New South Wales. So you can see that most of the farm we have actually surveyed in Broadwater, Condong, and Howard Mill area. They have medium to high nematode hazard index. That means that they potentially incurring yield loss. So we have found similar result in Harvard. So you can see that the hazard index are within medium to high. So, but if you look at in terms of actual nematode, for example, in Harvard, you can see actually the main reason the hazard index is very high because of the root lesion nematode is very high in numbers. So they are already in damaging numbers in almost every field. So we have this problem. So we actually reestablished again that nematode is a problem in our surveyed area. And then what can we do actually about that? So there are lots of work been done during last 20 years about under the auspices of the Yield Decline Joint Venture Project. And then we're just going to discuss some of these. So one of the things came up as a crop rotation and fallow. Why we do the crop rotation and fallowing? The reason is that nematode is a obligate parasite. So without the host, they can't actually live. So nematode actually, so if you have a crop rotation for 3 months, then nematode will die. So then you can actually plant it, and then you think that that should be all right, and then you can actually grow the plant happily ever after. So, but that's not the case, because the nematode, they die, but the nematode is not actually die. So they actually lay eggs. The eggs can actually live in the soil for a long time. So what's happened when you actually plant sugarcane, it's actually the egg hatch and they attack the roots. The nematode population actually come back again, the original number or higher number within 8 to 12 months. So it is a very short time control. Chemical control, yes, there is only one chemical actually registered in Australia for this pest. But the problem is chemical control is expensive, and you know that this is not environmentally that safe. The other problem with, I think this is the main problem with chemical, if you actually work with nematode, this chemical, you will find that actually they are very erratic. That means that if you have a sandy soil, lots of nematode, you can apply the chemical. You can actually get better control because of that. They can actually disperse within the soil evenly. But if you have a clay soil, it's very hard to actually get control in the clay soil or in the heavier soil because of the dispersion issue. So they are not very effective in the heavy soil. Also, that actually depends on other soil factor and the environmental factor. You need actually rain right after the application or irrigation right after the application. So there's another thing been done last, under the auspices of Yield Decline Project, which they found that soil organic matter, trash blanket, or high level carbon create suppressive soil, which is true. But the problem is that how? So it is hard to establish. So there is a huge knowledge gap in this area. So this is the main issue. So what about the biological control? So there are some preliminary work being done in different area, and biological control may be effective. So that's what I'm going to talk about these things a little bit later. And then we have the resistance variety, which could be nice if you have. Unfortunately, we don't have resistant variety, particularly for lesion nematode in Australia. So these two actually, this biological control and resistant variety are the main talk of this presentation today. So biological control. What are the biological control? It is the way of management of nematode by introducing the natural enemy or predators. So in case of nematode, usually we found that there are lots of fungal and bacterial biocontrol which can actually reduce the nematode number. So you can see in the top of the picture, there is a fungal nematode trapping fungi. What it does actually, it produces ring-like structure in the soil where nematode actually go through the ring, it collapses, it's almost choke it, and then it's actually colonized within the nematode, so the nematode dies. So the second one is bacterial. You can see the nematode infested with bacteria. One of the bacteria is important. We call it Pasteurella. I'll be talking about these things just right on. So Pasteurella. So what is Pasteurella? So Pasteurella is a gram-positive bacteria. It produces endospores, which means that it produces shell around this in a certain situation, which actually protects it from range of environmental stress, like if you heat or toxic environment, it can still sustain. So the other important thing about the Pasteurella, it is very host specific. So that means that, for example, the Pasteurella penetrans, it only attacks the root knot, not other nematode. That means that it doesn't attack the free living nematodes or other nematodes. That's why it's actually very popular. And the most important thing is there are lots of commercial interest in developing Pasteurella biocontrol product. So for example, Syngenta developed a product called Clariva, which is currently being marketed for the control of soybean cyst nematode. So this is the picture of Pasteurella endospore. You can they actually all look like flying saucer, a bit scary. So they stay in the soil, and when the nematode, this is a juvenile nematode of root knot, when it actually move through the soil, it actually attached on the skin of the nematode as like a Velcro. And then after that, it produces some kind of mycelia thing, and then it actually grows inside the body of the nematode. So we have done some work. So we wanted to see actually what the situation of Pasteurella in sugar industry. So we developed a research project, and this to actually assess the distribution of the Pasteurella penetrans, which actually parasite of root knot nematode in Australian sugar industry. And then we wanted to see whether these Pasteurella present in the sugar industry are able to control the root knot. And then last thing, even it's able to control the root knot, how much Pasteurella we need to actually effectively control the root knot. So these are the results. We found that about 30% of the field are infested with Pasteurella, but saying that only 3 fields out of the 126 been surveyed has relatively high level of Pasteurella, which can actually control the nematode. So the other field, they don't have actually, we found very like a scattered or very not that many. So what we did actually, after that we set up a glasshouse trial. And then we get the Pasteurella infested soil from the field, and then what we did, we autoclave some of the soil, and then some part of the soil actually, some portion of the soil actually remain untreated, and we set up a potting trial in the glass house, and then we planted sugarcane plant in the pots, and then after that we inoculated them with root knot. You can see after 33 weeks of inoculation, the Pasteurella in the autoclave soil, that means that the soil which we already killed all the Pasteurella, has about 200 times higher Pasteurella, in the nematode compared to untreated one. That means that the Pasteurella present in the natural soil has the ability to control the nematode in Australia. Then we looking for the actually concentration, how much concentration of spore we need to control the Pasteurella, to root knot. So we tried from 6,000 to 50,000 spore per gram of soil. And you can see that, so they can, regardless of 6,000 to 50,000, they actually significantly control the, reduce the number of nematode in treated plants compared to the untreated one, about fourfold reduction. So this is as people say, that picture says 1,000 words, and then you can see that in the left there is a Pasteurella, root knot infected roots, but in our pot trial, and then you can see the Pasteurella treated root knot infected roots on the right. So you can now understand why Pasteurella is so popular. Now, then comes to the resistance. So before going to the main topics, I just want to say that the resistance, what is the terms sometimes we use susceptible? What is the susceptible means? The susceptible means the plant, in terms of nematode, the plants which support the multiplication of nematode in the roots. When the nematode attack the roots or they enter into the root, it doesn't actually mind to actually grow the nematode on the root. But in case of resistant, they don't actually support the multiplication of the roots, or they actually resist the multiplication of the root. Sometimes actually even the nematode can actually attack the root, they can go inside, but they can't actually reproduce. Another term we usually call tolerant. What is tolerant? The tolerant is the plant can still grow and then give, and then they don't, without losing the productivity in presence of nematode. Even they actually attacked by the nematode, they still maintain their productivity. Then comes intolerant. The intolerance means that plant can suffer even with a small attack of nematode. So we sometimes could be hyper sensitive or hyper resistant. So as I mentioned that the breeding for nematode resistance is another way of way of controlling, we are exploring. So, but the problem is that we don't have any nematode resistant variety in Australia, or we can say the nematode resistant parents. So we need to find out the gene which from somewhere else. So that's what we wanted to see, actually, whether the wild cane can be brought into our breeding program to introduce the gene which could be actually resistant to nematodes. So the first objectives of our work was to identify the sources of resistance in wild germplasm like Erianthus or Spontaneum, and also the crosses and make crossing and also test the progeny of this integration family to see whether they are resistant to root knot and root lesion nematode. And then if there is a resistant, then we can use them as a parent to introduce the resistant gene into our breeding program. And then if some looks pretty good, we can actually use them as a commercial variety. And then also you can use existing marker information into the integration family to identify the potential marker for the resistant gene. So, as I mentioned before that we started this work about 10 years ago. First work been done from 2011 to 2014. What this work been done mainly on the integration progeny originated from China in the glasshouse. And then from 2014 to 2016, the further screening work been done on the integration clones in glasshouse. Also, we took some of the promising clones which showed resistance in the glasshouse in the field to the field to see actually whether they are resistant to root knot or root lesion nematode in the field condition. Just the main thing actually, we want to actually see the reliability of our glasshouse trial in the real world. And then we also screen our commercial clone, see the degree of resistance of this clone to the nematode. And from 2017 plus, we actually screen more our basic germplasm population from our SRA collection to see actually how resistant and root lesion nematodes. So this is the one of the result from our earlier trial. So you can see the on the x-axis there's all the cross type, and then the y-axis this is PI value, we call it reproductive value. What does it mean? That the PI is the initial nematode population used for inoculation in the glasshouse trial, and then PF is the final population after harvesting. So that means that if PI value is high, that means that actually is more susceptible. If PF is low, it's resistant. You can see from this graph that and these are from the those colored indicates the basic F1, backcross 1, backcross 2 up to backcross 3, and then if you look at the basic clone of spontaneum and erianthus, they are actually more resistant. And then if you go into the advanced progeny, the population actually more or less susceptible, or we can say more susceptible. But and these are our commercial clone. You can see they are actually almost all susceptible. And this is another basic clone, Erianthus proceras and Erianthus, Saccharum robustum. They're actually all susceptible. So, but even if you see that they're in Saccharum spontaneum and Erianthus, they have more susceptible progeny in the advanced stage as advanced progeny population, but still some of the clone from this advanced population actually has lots of, they showed resistant to root knot and root lesion nematodes. So this is this graph for the root lesion. As you can see, they actually kind of similar trend. So I'm not going to talk about this more because this is kind of a similar trend. So this is the trial as I mentioned that we wanted to see the progeny which are resistant to root knot and root lesion nematode. We took them in the field to see whether actually how much they're resistant, whether our result in the glasshouse actually are reliable. So these are our integration clones. You can see on the left side and on the right side, and on the left side our commercial varieties. So this plant actually grown for 3 years, and then you can see the nematode population, root knot nematode population actually remain or significantly low within the integration population. But if you look at the commercial variety, they are not that good. So after plant crop, they are all took off. But saying that we don't have much luck about the lesion nematode, because there are some resistant, but they are not that consistent in the field. So what we did actually, we wanted to go back to the basic, and we screened all our spontaneum population to see actually how much resistance we got for root lesion nematode. So this is the result we can see. So this shows that so this is the root lesion nematode population from our SRA collection. So these three actually not saccharum, so they are erianthus, and the rest of the clone are saccharum spontaneum. You can see, and these two are our commercial clone, so commercial variety. So you can see that we have significantly high resistant compared to the commercial clone in our saccharum spontaneum population. But if you look at the root knot, they actually, most of they are immune to actually root knot nematodes. So I guess we see that there is a hope. At least we can actually use them for future crossing. So now, as I mentioned that these are my last few slides. Soil health indicator. How can actually use nematode community as an indicator of soil health? As I mentioned that nematode, they are the vital contribution to soil health through the movement of the nitrogen and carbon in the soil, and also they are essential part of the soil ecosystem. So that can be used as an indicator of soil health. So why actually we use the nematode as an indicator of soil health? There can be some other way you can actually measure the soil health. You can use the total organic carbon, labile carbon. You can use the like enzyme activity. You can measure the carbon dioxide. So there are lots of ways you can actually measure the soil health. But what's the benefit actually measuring soil health using the nematode? So I'm just showing you one of the, so as I mentioned that the nematode is a very good colonizer. They are almost everywhere. But saying that, they're actually most diverse community in the earth. So but the nematode in one place or one environment is not same as the nematode in the other environment. So that depends on what type of nematode you will have. It depends on actually what is the environment of this particular soil or this particular place. So that means that, so we use this formula, we call it colonizer and persister type nematode. What does it mean? That when there is a disturbed soil or lots of disturbance or organic matter, and cutting the tree and plowing out, you will find lots of organic matter. And there is a then you actually this system become much more bacteria related or bacteria, and then you will find lots of bacteria-voral nematode, bacteria-vorous nematode, which actually predate on bacteria. And then when the systems come sort of bit of a slows down, and then we found another types of nematode we call is CP2. And then when actually we have the trace blanket and we have minimum tillage, you will find actually another types of nematode. But when you're in the like in rainforest, it then remain undisturbed for hundreds of years, you will find different nematodes. So just looking at the nematode, you can actually identify what type of soil this one and what sort of stress they're actually going through. So let's move on. So you can use different indexes, about 20 indexes you can use to actually do the soil test to actually measure the soil health. So this is one of the tests that we have done. So this is the result of the Harvard survey work. So we measured the nematode maturity index or soil maturity index, which, so in general, if the maturity index is below the 3, that means that the soil is like a disturbed soil or depleted soil. But if it's actually more than 3, the soil is a mature soil. It's got a nice soil food web, and it's got so this is the like a much balanced soil. So we can actually put them in a different way. So this is we call it CP triangle, which is much easier. Actually, we can visualize the problem. So what does it mean? So in this graph, it shows that if you are actually in this position, you are in a enrichment condition. That means that there are lots of bacterial parasite, bacteria-borne nematode. But if you are actually in the left corner, they're actually more stable. They're actually more, this nematode population shows that they're actually in a stable situation. But in our result, it shows that actually most of the sample we collected from this field, they're actually in the stressed. So you can see this can be used to monitor the soil health, which is in a good sense is good actually. So the grower can say, okay, I'm in here, how can I actually get into there, not into that side. So the other option, I'm just sort of shortly, so we can use nice little call is radar chart. So these are our result. It shows that the nematode, the soil actually have heaps of herbivore. That means that most of them actually plant parasitic rather than other nematodes. So what should be the like a typical perfect condition? So usually if it's a perfect soil, it would be like that. So it will be omnivorous nematode, it will be predator nematode, and there will be fungi-borne nematode. So there will be much more balance. But in our case, it shows that the nematode, the soil is stressed, as well as it has lots of parasitic nematodes. So what is the take-home message for us? So we found that majority of the sugarcane farm had high or moderate or high number of parasitic nematodes, so which actually potentially impacting our productivity. And for root knot nematode we have a potential biological control, but there is a big knowledge gap. Still lots of work needs to be done. Sugarcane integration lines, they are resistant to both types of nematode, but they're actually more resistant to root knot. Saccharum spontaneum, we found actually they have high level of resistance to both types of nematode. So as plant breeders, they produced a population by crossing Saccharum, a highly resistant Saccharum spontaneum with Q208. This population will be screened for nematode resistance in 2021. And the last one, as I just mentioned, the community analysis. It is a potential biological soil health indicator, can be used for monitoring the farming practices. So in conclusion, I can say that nematode pose a complex soil health issue. We need a multifaceted strategy to manage the nematode. There is not a single silver bullet. So the number one, we can say if we can develop resistant variety, and then if we use this improved soil health, and so that could be so then we can actually manage the nematode successfully. The other thing about actually the root nematode resistance, especially for root knot resistance, because some of the high value crop like soybean and vegetables crop, they are actually highly susceptible to root knot. But if you have a root knot resistant variety, you can actually use as a rotation crop to actually grow the susceptible other crop. And as I said, the biological control, there is a future, but lots of work needs to be done. Thank you very much, and thanks for listening. Thank you.
Thanks, Shamsul. Are there any questions for Shamsul? So we've got Salam saying, how do you relate nematode numbers in the soil health to plant? How do you relate nematode numbers in the soil to plant resistance or susceptibility? Was from Salam.
So I think the soil health and susceptibility is a different thing. Actually, the soil health is actually condition of the soil. So we use the nematode community to measure the soil health. But with the susceptibility, as we said, that we don't have actually resistant variety in particularly for root lesion nematode yet, so we can't actually help it. But without this, if we have this resistant variety, and if we actually test the soil, and then we see that there is a problem, then we can recommend then actually for use resistant variety.
Okay.
Yeah, sorry, Shamsul, my question was not about soil health.
Okay, so yeah, what was your question?
Yeah, how do you relate nematode numbers in the soil to plant resistance or susceptibility? That's the question.
Oh, okay. So if we have a more like a root knot or root lesion nematode, then if we have a resistant variety in hand, we can actually recommend like some other, like for Pachymetra and others. So at this moment, it's a good question. We don't actually have any.
Does that answer the question, Salam?
Well, I think I might ask another question then. You can have lots of nematodes in the soil, but the plant is not affected according to what you said.
Depending on variety.
Yeah. So counting nematode numbers now will not give you a good indication whether the plant is resistant or not. It relies on something else. There are so many other factors involved.
Yep, I guess you are right in some sense, but one of the thing is counting the nematode number. We haven't actually demonstrated that here, but Graham Starling did lots of work before. They found is actually the yield actually depend on the number of nematodes. So if you have a high number of plant parasitic nematode, it definitely affects the yield. So it doesn't actually, but the yield loss actually depend on many other factors as well, as I agreed. So if you have a very good condition, even with Pachymetra or other, if you have very good condition, you have good fertilizer and management, your yield loss will be less, I guess.
Okay, so going to Nick. Nick, do you want to ask your question?
I just also want to just quickly, Salam, maybe is your question relating also to how the resistant varieties are, how we do the screening? Is that also what you would like to know?
Yeah.
Okay. Yeah. So Shamsul, maybe you could explain how the resistant screening is done, because that's different than assessing it in the field.
The resistance screening been done, actually, we look at we inoculate them in our glass house, and then we grow the plant, the test clone, and then after 12 weeks, we harvest them, and then we look at actually whether the nematode number actually grow in the roots. So if they actually grow in the roots, that means that it's actually supported more nematode that actually has the potential impact on yield. If there is a not much increase in the roots, so we can say this is resistance. So that means that at least for nematode, it wouldn't actually do any damage. Does it answer your question?
Yeah, we're getting quite a few questions. So Nick, can you ask your question, please?
Sure, Shamsul, my question's very quick. Has there been any look at the population structure underneath the resistant or tolerant varieties as parameter?
I don't think actually anyone has done, but what actually Graham Starling's research show that actually if we no, I think no, it's not. Sorry.
No, that's okay. Thank you.
No, not been done.
Okay. Harjeet, you have a question. Would you like to ask your question?
Yeah, I have been reading this thing about Graham Starling's 80 million per annum thing at quite a few places. How did we arrive at that number and saying this is what it costs the industry? When it's so hard to find the numbers and associate it with resistance or susceptible varieties and how much impact it has on a susceptible variety versus the resistant variety and how much nematode pressure is there around the world. Do we actually know what's the cost of this problem to the industry?
Yes, I think the Graham Starling did. Do you have the paper of Blair and Starling or Blair in 2007? They did actually substantial work on the survey using the nematicide. So they actually use nematicide substantially, and they actually find out the yield loss, and they calculate the loss of yield based on their test. So I can send you the paper if you don't have this. So have you read this paper, or you want me to send it?
No, I do remember the project that we had funded on the introgression clones that was looking at the impacts and whether in field you can actually test whether the clones are resistant or susceptible. And I know that project didn't come up with any conclusive data around that. So do we have field-based data actually?
Yes, there is. Yeah, that's what I just said. For our project, we don't have, but under the auspices of yield decline project, they have actually field-based data, and this shows that it cost more than 80 million. So they did actually a very massive, like a test Australia-wide, and they found that actually they cost. So that's the figure they came out. And for our trial, actually, I guess there are some issues. Number one, actually, for spontaneum, we didn't have actually much resistance in the field. So even in the glasshouse, we were sort of struggling to finding resistance, but we had some good resistance for root knot. So that's what actually we didn't actually, and the project actually unfortunately didn't get actually funded further to actually test in the field. So
yeah, so that's what I'm saying, that Sterling collected some data based in past based on the varieties he had that time, and that's what the calculation was done. Today, whatever varieties are being grown out here based on the resistance susceptibility levels of those varieties, today do we know what does it cost the industry?
So I think the number one thing is that we don't have any resistant variety. So this is full stop. Because in the glasshouse trial, we actually didn't found any variety currently in Australia that are actually resistant.
Yeah, but we don't, I think what Hajit's asking is, do we have data on today's losses? And I don't think we actually have data on today's losses. But there could be assumptions made.
Yes.
So we must keep moving. There's quite a few questions coming through. So Sanaz, do you want to ask Shamsul your question, please?
Thanks, Shamsul. That was a great presentation. I just wonder if you have ever looked at the different control strategies from economic perspective. Have you ever compared them, the cost effectiveness of the mentioned strategies?
No, actually, we haven't actually. So we only thing actually we have done last 11 years, sort of like on resistant variety or sort of finding out the resistance. I think that would be very good if we actually have some economic analysis done. Now, at least now, we have an economist on board, I guess.
Okay, but so you have no idea. So how you can compare the strategies, or in your opinion, which one would be the best strategy?
Exactly. Yeah.
I think just from my experience in other parts of the world, if you are looking at reducing numbers of nematodes, and Shamsul was alluding to this in the first part of his presentation, it's really that you need a variety that's got resistance, so that nematode numbers are reduced over a period of time. I know in Africa, they talk about 3 years for root knot nematode. So if you look at our crop, we get at least 4 years out of it. And if you're looking at a rotation crop with other crops, or even going back into cane for that matter, then you get significant control. So Sanaz, you also wanted that paper. So Shamsul, can you share that with everybody, please? That paper
Yes.
You were talking about from Sterling?
Yes, I'll do that.
Okay, Phil Jackson. Phil, would you like to ask your question, please?
Hi, Shamsul.
Hi,
just
Phil.
wondering, with that Spont by Q208 cross, are you thinking about trying to identify markers or QTL from that Spont resistance source?
Yes, actually, we're talking with Karen about this, and we did some actually test in our glasshouse under a project, like a research award project. So yeah, we're trying to actually, because we found actually this is this particular clone, which is SES208. I think BES208, which is very resistant and also is at very high root and shoot biomass.
Okay.
Thanks.
So Brad's also put up he's put a link there for the paper.
Thanks, Brad. It's already done.
Yeah. And I think, are there any other questions from anyone else?
I guess I'm going back to Harjit. I guess, yeah, you are right. So we don't actually have much data. So all the data actually 20 years old. And you are absolutely right. Actually, we don't have information on the current variety. Actually, how much damage we actually having. So I guess that this is another thing. Maybe research funding panel can actually help us to actually do some more survey and do some yield loss trial.
We've got one more question from Nalini. Thanks for the presentation. I'm interested to know the pathogenic was calculated.
So you're talking about the hazard index?
Would you, sorry, I've got pop-ups here, so I can't get to the question. Nalini, would you like to put your question across, please, in your words?
Yes. Thanks, Shamsul, for your presentation.
Thank you.
I'm interested to know the pathogenic that was calculated, the nematode pathogens. An example you have showed in this slide that the hazard factor you have showed there's the value was there 1 and 0.2, something like that. So I want to know how that is being calculated.
So we actually, it's on the assumption of the, on the basis of the pathogenicity. So we found that two most important nematodes are root knot and root lesion nematode. And root knot actually, in particular, most destructive pathogen. So in particular in sandy soil. So what we did actually, we converted the pathogenicities, we sort of we then actually, this method, we use a relative pathogenicity of other nematode in relation to root knot. So that's what actually, if we have a highly pathogenic nematode like root lesion nematode, it is a very damaging, but it's not actually as damaging as root knot. But if you look at actually how we actually going to get the number from, so this is actually, we had a discussion with Graham Starling, who had a 30 years of work on sugarcane nematodes. So this is actually sort of all our discussion, we find out these numbers, but it's sort of like an assumption on the basis of the experience. Does it answer?
Thank you.
You're welcome.
Thank
Marie,
you.
you've got a question. Are there differences in the infection rate of roots by root lesion and root knot nematode? Is that right, Marie? Is that your question?
Yes. I think the root knot and root lesion, they actually they have a different space. So if it's a very sandy soil, definitely the root knot will be a more prominent one. But in other soil, you won't actually see that much root knot. So it just depends on what type of soil you actually, but you will find actually more or less root lesion nematode in sandy soil, but we found actually more dominant one always root knot in sandy soil.
Okay, and then Helen's got a question. Has the metabolic footprint for the different types of nematodes been used for crops other than sugarcane? Do you have an answer for that one, Shamsul?
Yes, of course. There is lots of I think that they've been done with most parts of horticultural crop. Even they've done for soybean, and I can send you a few example if you want. So yeah, they've been done for other crops.
Okay, excellent. Well, guys, we've run out of time. Thank you very much, Shamsul, for your presentation. This is going to be recorded and will be on the SRA website and in the next e-newsletter. So thanks very much, Shamsul, and thanks for everybody who participated and dialed in today. Thank you.
Thank you. Thanks.